Hey everyone, this is a short video reviewing some of the essential content from our lab on cell structure, function, and the cell cycle. So let's dive right in. You do need to know all of the major organelles and their functions. There is a list of them in your lab manual, and those are the ones that you should focus on. This diagram right here is actually from Visible Body, so you should be able to take a diagram like this and recognize and identify each of the major organelles, and you should also be able to describe the primary function for each of those organelles. And if you pop into Visible Body to look at this diagram, you can actually rotate it around and you can click on each of the organelles to get a description of the organelle and its function. And another place you could get that information is also in your Martini textbook for the lecture part of the class. So there's kind of multiple places you could get the information about cell organelles. Also, be aware that you could be asked about these organelles on the practical exam with a model like the one from Visible Body, or the more simple diagram in your lab manual, or one of the cell models that was in the lab. So you want to make sure that you're comfortable enough with all of the organelles in different models and diagrams to be able to recognize, identify, and describe them. Okay, for the cell cycle, I popped in the diagram from Visible Body here, which is very pretty. And then there's also the cell cycle diagram that's in your lab manual. I would suggest that in your lab manual, you go ahead and you take notes in the space sort of around this diagram, indicating what's happening in each of these major stages or phases of the cell cycle. So, big picture in the cell cycle, right, is that we have interphase, which is made up of G1, S phase, and G2, followed by M phase or mitosis, which will consist of prophase, metaphase, anaphase, and telophase. We will likely have cytokinesis in most situations, leading to two new cells that will then enter G1 of interphase. And if we look here, we can see in our Visible Body model, they talk about G1 phase and how the cell is going to be growing and duplicating or increasing the number of organelles. It will be making lots of proteins. In other words, it's doing its job, whatever its job is. If this cell is destined to undergo mitosis, then it will shift into S phase. And S phase is where all of the DNA is replicated. So every one of your 46 chromosomes will get copied during S phase, so that by the end of S phase, we will have 92 chromosomes inside that cell. And then the cell will enter G2, which is a phase where it will continue to grow if it needs to, make any more organelles if it needs to. But really, it's waiting for the proper signaling to indicate it should, in fact, enter M phase and take those 92 chromosomes and divide them equally and identically into two nuclei, followed by cytokinesis, which would result in the splitting of the cytoplasm and ultimately the formation of two cells. All of these phases are essentially governed by checkpoints that determine whether or not the cell should in fact progress through that phase and continue into the next phase, or if there are any major issues that the cell might apoptose, in which case it will self-destruct to prevent passing on any problematic DNA, for example. There is additional information about mitosis in Visible Body. There's a great little video and some surrounding text. There's also information in your Martini text. And don't forget, there are PowerPoints in Blackboard in your lab course that provide information as well. Okay, for our M phase, so let's just go back here. Here's M phase right here in our cell cycle diagrams. Now, what we needed to look at in the lab was what those cells looked like under the microscope and be able to recognize them. Are they in interphase? Notice that when the cells are in interphase, the nucleus is just kind of a dark spot. There's nothing that stands out about that nucleus. We can see it has a nuclear membrane. We can see the cytoplasm. We can see the cell membrane. And that's really all that we can see in those cells under the microscope. So as you can see over here in this little description from your lab manual, cells will spend most of their time in interphase. We can't see the chromosomes because they haven't condensed. And that's sort of the hallmark of interphase. But as we progress into M phase, we're going to start in prophase. And the hallmark of prophase is that a few major events are going to happen. Number one, our chromosomes are going to condense down and become visible, as we can see right here. And the nuclear membrane is going to start to disintegrate and disappear. We are also going to see a replication of the centrioles, and they will start to move apart and form those spindles between them, creating that kind of star-like aster. And the spindle formation will develop, and they will move to opposite parts of the cell. And so we can start to see that the chromosomes will start to attach themselves to the spindle formation via their kinetochores. And once those chromosomes have aligned themselves down the axis or the center of the cell, we will have shifted into metaphase. So there are sort of, early prophase and late prophase might not look identical, but metaphase is a real giveaway because they're going to be lined up right down the middle like we see here in these cells. Then we progress into anaphase, which is where the paired chromosomes will separate so that one copy goes to the left, if you will, and one copy goes to the right. And that is how we're going to create identical cells at the end of this process. And so if we look at this picture from a microscope, we can see that the chromosomes have pulled apart and are moving down the spindle formation towards their centrosomes or centrioles at opposite ends of the cell. And by late anaphase, we may even see a cleavage furrow start to develop. And then as those chromosomes reach the opposite poles of that cell and we see a significant cleavage furrow and the spindle formation starts to break down, we have reached telophase. And when the chromosomes have really settled in at their respective poles, they will start to uncondense or unravel and become thread-like again, and we won't be able to see them. And cytokinesis will progress, assuming this cell is going to undergo cytokinesis. And again, most cells do. So we'll see a deep cleavage furrow that will progress into complete cleavage until a cell membrane is formed between the two poles, creating two separate cells. And here's just another set of images with a cartoon version and with what those cells look like under the microscope. So you can kind of familiarize yourself with what they might look like with different stains or different microscopes kind of thing. Take note that I will say I don't love this example right here of interphase. There's a little bit of what to me appears to be condensed chromosomes here. This is a beautiful prophase, but this to me looks more like very early prophase than a true interphase, but it's hard to tell without seeing the whole slide of cells and being able to compare them. Okay, and here are a few images that are similar to what we saw in lab. The cells are often kind of faint, but you can see here's a prophase that has that dottedness that is pretty typical of early prophase. We can see chromosomes lined up down the center of the cell and the spindle here with the centrosomes on either side. Great metaphase. Here we can see the chromosomes just pulling apart. So this is a very early anaphase, quite nice. Here's our centrosomes, and we can see the spindle formation as well. You can see the cell membranes around the cells and the cytoplasm. So this is pretty typical of what we saw in lab under the microscope. Here's a couple more pretty examples. This is a great telophase. We can see cytokinesis is complete because we can see the membrane down the middle. We're even starting to see these chromosomes are starting to unravel. And within, a few minutes, if we could move the clock ahead, we would start to see the nuclear membrane reform. Over here, we have some beautiful metaphase examples. And down here, we also have another early anaphase. So some great examples for you to look at and just recognize. So hopefully this helps you feel a little more confident about mitosis and what you need to know for your practical exam.